Great. So welcome everyone to our next seminar in the LIBRE Hub series. In this project, we focus on expanding access to open hardware research tools for bioimaging. And so we'll hear about the research of Dr. Nathan Lord. He's an assistant professor at the University of Pittsburgh, and he'll talk about optogenetic control of nodal signaling patterns. So thanks so much, Nate, for joining us, and we're super excited to hear your work.
Awesome. Well, yeah, thank you very much for the invitation and for the opportunity today. This is really a lot of fun, and hopefully we can get into some of the technical weeds, which I think would be a lot of fun to talk about. So basically the structure of the talk today, we come at sort of doing optogenetics and imaging, motivated very much by a set of biological problems. And so to give everyone context for the story in this sort of like first paper from our lab that I'll talk about, I'm going to spend about 5-10 minutes talking about the biology that led us there, and then we'll get into kind of how we're doing the instrumentation and how it was built and how we apply it to these problems. And so if you bear with me through the biology for a few minutes, we'll get into the sort of cool technical stuff after that. Great. So I thought I'd start off with a picture of a zebrafish embryo that's about 4 hours old when this was taken. And this is our organism of choice that we'll be talking about today. And so all the different colors that you can see here mark sort of the earliest stages of cell fate acquisition for this embryo. So even though it was only born about 4 hours ago, and at the onset of its life, all of the cells would have been like more or less identical to one another. They've already started to sort of figure out where they are inside of the embryo and then make decisions about who they're supposed to be when they grow up. And the sort of at a 20,000 foot view, what my lab is interested in understanding is how is it that the embryo communicates instructions to each one of these cells. And we have a special interest in understanding what happens when things go wrong, because it turns out that there are all kinds of interesting physical considerations that make it quite a hard problem to communicate accurate instructions to all these different cells. And we think that embryos have ways of kind of correcting course when things go wrong. This is something that we're studying actively in the lab. So we come at this in a bunch of different ways in the lab. In very broad terms, we break this up into two main classes of problem. The first one that I'll call pattern generation. And what I mean by that is we want to understand how, networks of signaling factors and proteins and genes inside of the embryo conspire or come together to create spatially resolved patterns of gene expression or signaling, which ultimately tell the embryonic cells what they're supposed to be doing. And the second part of the problem we'll call pattern interpretation. And what I mean by that is once you have a pattern, how do cells read it, extract information about what they're supposed to be doing, and then use that information to construct some kind of a tissue with a well-defined structure. And so the story I'm going to tell you about today basically started with our work on the first problem. So where do patterns come from in the zebrafish embryo? And then kind of led into a set of tools that we're going to use, looking forward to understand how patterns are interpreted. So to just lay the groundwork for pattern generation, I think that the simplest way of creating a pattern that cells can use for instructions in an embryo is probably pretty familiar to most people if you've ever had a developmental biology course. The basic idea is to use a morphogen gradient. And so just to sketch that out, what it means is you have some kind of a molecule that activates responses in target cells. And so here in this little cartoon, we have that activator produced in this special cell in the middle that serves as a source. And it secretes this molecule into the extracellular environment where it diffuses away to form a concentration gradient. And so cells can effectively use this concentration gradient as a coordinate system. So cells that see lots of it are close to the source. Cells that see less of it are further away from the source. And then by sensing how much of that signal they have in their environment, they can sense their distance from the source and therefore where they are within the embryo and what they're supposed to be doing. But the observation that kind of got everything I'm going to tell you today into motion was that if you look at natural patterning systems, they are rarely this simple. And one elaboration that you often see is negative feedback control. And what that means is the activator, in addition to driving responses in the target cells, also drives the production of inhibitors of its own signaling. And so this is, at first blush, kind of a paradoxical thing. Why would an activator want to both turn its pathway on and off simultaneously in these cells? But you can imagine different things that this could be useful for example, restricting signaling in space, or maybe turning it off when it's not needed anymore. But of course, because biology is biology, things can get even more complicated than this. And another elaboration that you often see is that the activator, in addition to driving responses in target cells and production of its own inhibitors, also turns itself on or induces its own synthesis. And these kinds of interlinked feedback systems, if you get the biophysics just right, can do all kinds of really cool biochemical or dynamical backflips, like forming, spontaneous stripes or spots or traveling waves, all without the need for external information. And so really, there are a lot of things that you can kind of imagine feedback doing in embryos. And the overarching question that motivates everything I'm going to talk about today is, can we get our heads around what feedback is doing in a specific context in zebrafish development? And that context is nodal signaling. So nodal is a TGF-beta family signaling molecule that orchestrates one of the first cell fate decisions in vertebrate embryos, which is which germ layer to join. So in a fish embryo at 4 hours after it's born, you've got cells on the top and then yolk on the bottom. And at the boundary between these two things, you get production of nodal ligands. And then they diffuse into the extracellular space in the cell cap, and then they form a concentration gradient. And the thinking goes that cells that see lots of it become endoderm, cells that see a little bit less become mesoderm, and cells that don't see any at all become ectoderm. So that would be the sort of the gut tract, the muscle and connective tissue, and the skin and nervous system. There are a couple of molecular details that are useful to have under your belt to understand what we're going to talk about today. So nodal is this dimeric extracellular ligand that works by binding to cell surface receptors. I just want to call out this protein OEP. This is going to be important for a couple of the things we're going to go over today. So OEP is a member of the receptor complex. And the important thing to know is that if you don't have it, nodal cannot bind to the cell surface. So it's like an obligate, we call it an obligate co-receptor. Nodal binding to the receptor is antagonized by proteins called lefties. But when nodal does bind, it turns on the phosphorylation of this transcription factor called SMAD2, which then goes to the nucleus and activates target genes. And in terms of motivating today's story, really the interesting observation is that if you look at the list of target genes that nodal turns on, you find nodal ligands themselves, as well as these antagonists, the lefty proteins. And the reason that's interesting is because that places the system into that most exotic of categories that we talked about before, where you have this interlinked positive and negative feedback loop, where if you put it in the right biophysical territory, it can spontaneously form patterns. And when I first got into this field, started my postdoc in Alex Shear's lab, this was basically the thought about what the system is doing. There's a really beautiful paper from his lab looking at diffusion rates and stabilities of these proteins and showing that it seems to live in precisely this territory where it could make these spontaneous stripes or spots. But from my perspective, the kind of puzzling observation is that if you take a look at what pattern this system actually makes in embryos by staining for the activated form of that transcription factor as like a readout of nodal signaling, what you see is you don't see stripes or spots or waves. You see a monotonically decaying gradient, which is strong at the source, weak at the top, and decays approximately exponentially. And the reason this is puzzling to me is because this is something that you don't need anything fancy to make. Like just diffusion and capture by cells is enough on its own to give you an exponential signaling profile. And so I'm really puzzled by why the embryo is using something so complicated and sophisticated, which I'll also mention is very deeply conserved, like all the way down to sea urchins, to make something so simple. And I've been spending the last few years of my life trying to pick apart why this might be. And I'm not going to get into this story in the full detail. We have a couple of papers published on this that you can look up if you're interested. But I just want to highlight one kind of puzzling observation that got us into doing optogenetics and trying to engineer this light responsive control of the nodal pathway. And that is if you look at this very particular mutant background, which is called a zygotic OEP mutant. So again, OEP is this obligate co-receptor protein that's required for nodal to bind to the cell surface. you see something really puzzling. And I just want to call out, for those of you who don't sort of study embryos, what a zygotic OEP mutant means is they have the egg of the fish has a little bit of mRNA from mom. It's deposited from the mother, but they cannot make any on their own because in the zygotic genome, the genes are mutated. And so effectively what that means is there's a finite supply of OEP, but they can't make their own. And so that's going to become important in a moment. But we can kind of look at a wild type embryo and you see that pattern that I just showed you where you have a gradient that's strong at the source and then decays as you go up towards the top of the animal. In a zygotic OEP mutant, you see one thing right off the bat, which is that signaling is weaker. That kind of makes sense. You know, like you have two sources of OEP and one of them is gone. So maybe you have like less signaling. But the funny thing is, if you increase the contrast to see what the pattern looks like, it's not a gradient, it's a stripe. So the source is still down here, but now you have this band of signaling that's about halfway up the embryo. And so we made this really subtle change and something, you know, completely unexpected and funny happened. And, you know, we went through a bunch of trouble to try to figure out why this might be. And I can just kind of cut to the chase and say that it's easy to explain with a really simple model. So if we have, imagine we have these ligands in the extracellular space and they can bind to cell surface receptors, you know, according to mass action kinetics. Once they bind, they get removed from the cell surface, and then cells put receptor complexes back on the surface with some rate to make sure they don't get depleted. That simple model can explain formation of gradient. So on the left here, we have the concentration of nodal receptor complexes, which is a proxy for signaling, and you see that you get the gradient just like you're supposed to. But the cool thing is if you set this K3 rate to 0, so you eliminate the embryo's ability to put receptors back, this is kind of exactly what's happening in a zygotic OEP mutant. They have some cache of receptor to start with, but they can't put it back after it's been consumed. Then actually what you get is something that starts off as a gradient. That gradient turns into a stripe, and then that stripe propagates up towards the top of the embryo. And this ends up being a pretty intuitive thing to understand if we step through what might be happening in the embryo. So, you know, at time 0, we have we sort of 1D the embryo into a source on the left and then a bunch of receiving cells on the right. The source cell sort of secretes nodal into the environment where it diffuses along. Initially, it gets captured by the cell surface receptors on the first cell, and you get signaling there. But then eventually those are consumed, and then the ligand can make it to the second cell until those receptors are consumed. And then it makes it to the third cell until those are consumed. And this just kind of plays out over and over. And if you think about where signaling is going to be, it's where receptors are occupied by ligand. And so first it's there, then it's at the second cell, then the third cell. And so this sort of depletion effect actually creates a traveling wave of signaling that goes towards the top of the animal. And if you look by immunostaining at wild type and zygotic OEP mutants, this is exactly what you see. So in a wild type embryo at 4 hours, you get a gradient. At 5 hours, it's still a gradient. And at 6 hours, it's still a gradient. But if you look in these zygotic mutants, first you start off with a gradient, that turns into a stripe, and then the stripe propagates up towards the top of the embryo in approximately a linear fashion. Okay, so the sort of end of biological interlude. Why did I go through the trouble of telling you about all this stuff, and why does it motivate the optogenetics? The reason is because these zygotic mutants, from my perspective, are surprisingly okay, even though they have this utterly transformed signaling pattern. So if we look at a wild type embryo that's got this sort of normal nodal gradient, this looks like a fish at 24 hours with all the stuff that a fish is supposed to have. In a mutant called an MZ-OEP mutant that doesn't have any of this functional receptor at all, so no gradient, you get a completely messed up embryo. It has effectively no endoderm or mesoderm. It's just kind of a bag of brain and skin. And these guys really do not make it much further past this point when this picture is taken at 24 hours. But the cool thing is, if you look at embryos in which you have a signaling wave, so these are these zygotic OEP mutants, so not a gradient, but a wave, they actually have a surprising number of nodal dependent structures. So there are defects to be sure. So anyone that studies embryos will know that will see that there are defects here, but you do successfully specify good somites, you get a node accord, lots of other nodal dependent structures. And so to me, the puzzle is how can you it's really interesting that you can utterly transform this pattern that nature seems to go to great lengths to create, and you can still successfully specify these really complicated tissues. And this really got me thinking, or wondering if, this graded signaling is necessary to successfully make a fish. So this model that I presented to you, we can call it maybe the classical model or the textbook model before that I presented to you before, makes a pretty compelling intuitive case for having a gradient. So, you need to have the signaling gradient because it creates this very precise ordering of germ layer precursors. And you want to have that precise ordering because it sets the stage for development later on. But the fact that, we can do so much with such a funny looking pattern suggests to me that maybe there's a lot more flexibility in this requirement than we would think. And so an experiment that I would love to be able to do is to, rather than having a gradient, just have a stripe of nodal signaling around the edge. And then maybe you get some weird mix of endoderm and mesoderm, and maybe that's good enough to do a lot of good development downstream. But the problem with this is this has been classically a really difficult thing to do because we can't just kind of paint signaling wherever we would like on an embryo. The sort of traditional toolkit gives us a lot of flexibility, but you can't just take a gradient and turn it into a stripe or flip the gradient upside down or move it. Those are very difficult manipulations. And so this got us into doing optogenetics, where we thought that if we could rig an embryo up so that we can shine light on it and get it to signal, maybe that could actually be this magic paintbrush where we can put signaling wherever we want and we can begin to address these questions. And so late in my postdoc and also early in my my independent lab, which we started here in late 2020, we've been building out this toolkit to be able to pattern nodal signaling optogenetically in live zebrafish embryos. And this involves some reagent development that I'll tell you about, as well as some instrumentation. So, of course, the first step is to engineer a light-sensitive nodal signaling pathway. And I want to be very clear that the strategy here that we used was inspired by this awesome paper from C.P. Eisenberg's lab in 2016, where they had this really clever trick to initiate signaling, where the basic idea is what you have to do to get signaling to work is to bring the type 1 and type 2 nodal receptors together. And the way that they do that is by tagging them on the intracellular side with these love domains that in the presence of light dimerize, but in the dark are not together. And so the hope is you shine light on them, they assemble receptor complexes inside the cell, and that leads to signaling. And this works really, really well. So this is taken from their paper. If you inject these receptor RNAs into fish and you shine light on them, you get sort of expression of this nodal-dependent transgene or nodal-dependent reporter gene everywhere in the fish in the light, but only where it's supposed to be in the dark. So the reason why we kind of got into reagent development rather than using these is because for the applications that I'm going to tell you about here, it turns out that the background activity in these receptors was a problem. And what I mean by that is that there's some degree of association in the dark that leads to productive signaling that for a lot of applications is not a problem, but for us was a problem. And so just to kind of illustrate that, if we inject nothing into fish embryos, this is what they're supposed to look at 24 hours. But if we inject enough receptors so that we can get induction of all the different nodal target genes in the light, in the dark, this is what the embryos look like at 24 hours. And this is, for people who don't look at fish embryos, this is pretty characteristic of what happens when you have an embryo with hyperactive nodal signaling. And so, because ultimately what we're interested in is trying to replace nodal signaling and look at phenotypes at 24 hours, this means that we just couldn't use these reagents to go forward. And so, in collaboration with Adam Cohen's lab and with his awesome student there, Harry McNamara, we started a project to try to improve these receptors. And we took inspiration from this paper that made optogenetic TGF-beta receptors to improve our background. And we made two changes. One was we changed the intracellular binding domains, or the photodimerizing domains, from love domains to Cry2 and CIVN for reasons that we can get into that are not super important here. But I think much more importantly, what we do is we take the type 2 receptor off of the membrane. And so the idea is that in the dark, now they're kind of sequestered into two compartments, and it's less likely that they're going to accidentally bump into one another and signal. But still, in the light, you can recruit the type 2 receptor to the membrane and then get productive signaling. And so the first thing to show is that this works really nicely, at least as well as the optoNodal reagents. And so this is just a power series showing in purple is just all the cells, and then in green are cells that are signaling in response to light. So you get more signaling as you ramp up the light intensity. That's the original reagents. And our new reagents, which we're calling optoNodal 2, we get very comparable dose or light responsiveness. And so we don't lose any of the activity on the high end, but importantly, you can inject the same dose of these receptors into the fish, and at 24 hours in the dark, they look just like normal fish. And so we were also able to dramatically reduce the background. A nice surprise that actually is a little bit puzzling. I have some ideas about why this might be, but we were happy to see, but we still can't completely explain, is that the receptors have much better dynamics. And so if we look at an experiment where we just give optonodal receptors a short pulse of light at time 0 and then look at signaling after that pulse of light ceases, what we see is they sort of turn on very rapidly, but then the problem is they just kind of stay on or continue accumulating. And so in our hands, these receptors behave a lot more like a switch where you shine light on them and they stay on for, approximately another 2 hours at least. That's about as far as we've looked. But our receptors, if you repeat the same experiment, you get sort of the receptors to turn on over approximately the same time scale, but then they turn off right after. And so by 110 minutes after stimulation, we have signaling that has returned approximately to background. And we think that this probably plays a role in, again, in why we don't see appreciable dark activity when we don't stimulate them. Great, so we have some receptors that work nicely and are quiet in the dark and strong in the light. And so now we can actually start thinking about spatially patterning embryos. And so we are using a platform that's based on a microscope that was originally developed in Adam Cohen's lab called the Firefly. And its purpose was to do like large format or large spatial area optogenetic stimulation and observation in brain slices. And this has some characteristics that are really, really useful for doing this in fish embryos. So the most important thing is that the field of view of the microscope is very large. In our current instantiation, it's about 20 square millimeters. And the reason that this is important is that fish embryos are huge. So like one fish embryo at the time that we are imaging is about 1 millimeter across. And so if you want to be able to explore more than just 1 or 2 patterns, being able to pack lots of them into one field of view is a huge advantage. And so currently, we're at a stage where we can get approximately cellular projection resolution, but get, depending on how we lay out the embryos, between 8 and 16 embryos per field of view. Since this is a crowd that's interested in open science, one thing that is in the supplement of the paper that anyone studying fish might find useful is we actually array these embryos regularly in space for patterning. And the way that we do that is by 3D printing kind of little miniaturized, like embryo-sized egg crates that we mold into agarose. And then it creates like a little indentation for each embryo that holds them still during patterning over the course of about, like 12 hours or so until, which is more than enough time for us to be able to pattern nodal signaling. And so those tools are in the paper if anyone is interested. But for the patterning, the basic idea is that we take a bank of LEDs and then we bounce light off of the face of a digital micromirror device, which sort of consists of an array of individually addressable mirrors, each of which you can think of as a pixel. And then we turn, the pixels that we want on to create an image, and that image is shrunk down to the size of an embryo, or more specifically to the size of several embryos, so that we can put signaling wherever we want them. And so with this pattern or this system in hand, we can, do funny things like define arbitrary masks, like a spot or a line or a bullseye, and then show that we can project those onto nodal embryos and then get it at the level of nodal activity. So around the edge here, what you're seeing, we're looking down on the top of an embryo. So the normal nodal signaling domain is around the edge at the equator of the sphere. And then we get the little spot at the top or the line across the embryo or the bullseye, if that's where we put signaling. And if you leave that signaling on a little bit longer, you can also get expression of target genes. And so this is a mesodermal fate gene. We can get the spot or the stripe or the bullseye. And then we can also get expression of genes in the nodal regulatory pathway. So this is one of those nodal antagonist genes, lefty 2. So you can get the stripe and the spot and the bullseye. And so now we're in a position where we really can kind of point at an embryo and project whatever kind of nodal signaling pattern onto it that we would like. So here's a slide I'm sharing just for this audience. So this is a picture of our current setup. The original Firefly was really built from the ground up from individual components. And for the sake of sort of expedience and simplicity, we actually built ours on the back of a sort of on the back of a commercial microscope from Mitex. So we use a Mitex Oasis macro microscope, which consists effectively of like a bank of LEDs that we buy from Mitex, which then you run via a light guide to a DMD projector. So I think getting good even illumination is one of the challenges with these microscopes. And I think in the Cohen lab, when this was done, completely custom, sort of required a decent amount of engineering to get that to work well. And so one of the nice things about using a kind of out-of-the-box projector is that those problems are effectively solved in the box, and we can just build around it. But so we have a TMD in here. The light is bounced off of a dichroic here and then down through a lens where you stimulate your embryos, and then we have a fluorescence detection path going up to a camera. So we modified the microscope in a bunch of different ways that are germane to the discussion today. So we added our own camera to it. We use a Hamamatsu Orca Fusion. There is no motorization of an OASIS macro, and so that was something that we wanted to change. So we added a motorized 50 millimeter filter wheel to accommodate the large field of view of the microscope. So we do like, I think 5 different imaging channels routinely. And then we added, to improve the efficiency of the detection path, we added a 4-band dichroic. And then we keep the original lens from the OASIS macro, which is a 2x.37 NA objective. We have tested the objective that was originally used in the Cohen lab Firefly microscope, which I believe was from Olympus. And actually, they seem to work pretty comparably, but the working distance on this one is a little bit higher, which is advantageous for our purposes. And then we use, again, for the projector, it's a Mitex Polygon 1000 DMD. And then to enable multiple position scanning, which is useful for a whole bunch of different projects in the lab, we added in a prior encoded XYZ motorized stage. So it's motorized in all 3 dimensions and encoded in all 3 dimensions, which is super useful. In terms of the open source stuff, so these are all sort of commercial components. We wrote the automation code or the front end for the microscope completely from scratch in MATLAB. And all of the components that that we are allowed to release, in cooperation with the manufacturers, we'll be releasing on a GitHub page. But basically, the idea is that we have MATLAB code that creates an object class for every piece of hardware on the microscope that moves or does anything, and then sets of class methods for each one of those objects, so that you can really think of it as kind of like scripting the microscope in just like a MATLAB file. And so every acquisition that we run in the lab is effectively a for loop where we're just stringing together actions for each one of these components. And we found that it just gives us lots of flexibility, and we can design arbitrarily complex experiments where we combine stimulation and measurement, and we're excited about the idea of kind of like interlinking those two so that the measurements motivate further stimulation and do closed loop control. But I think it's been really useful for us to kind of not try to use the out-of-the-box Mitex tools, but to build our own that allow us to really do anything that we want. And so that's the setup. Please, anyone who's interested, do feel free to ask questions. Happy to go into details. But the fun thing now is that now that we have the reagents and we have the microscope that lets us pattern, we can start actually doing cool biology. And we wanted to see if we could pattern, not just the structure of gene expression in space, but also self-fate decisions in space. And the first place we looked was in the specification of endoderm. So biologically, what happens in a wild-type embryo is we've got an embryo side face here. We have a nodal gradient, and looking down from the top here, we see the same gradient. And that gradient induces these scattering of endodermal cells, which go on to form the gut tract. And then so they're specified by 6 hours post-fertilization, but by 9 hours, they sort of crawl inside, which is the process of gastrulation, and they start to move in these really fascinating ways. And so what you see is the cells are sort of more spread over the animal, but also they are deep, meaning that they underlie all of the other cells, which is where the endo and endoderm comes from. And so our thought was, well, let's take an MZ-OEP mutant that has no nodal signaling and therefore no endoderm, and then project nodal signaling to where it's supposed to be in the animal, and then ask at 6 hours, can we see induction of endoderm? And at 9 hours, can we see it moving as it's actually supposed to do? So again, patterning actual cell fates and cell behaviors rather than just expression patterns of genes. And so this is what it looks like in a wild type embryo if we collect embryos and then stain them for these fates. So here's there's the expression of the endodermal marker gene called SOX32. So sort of ignore this big massive green below the cells. That's actually the yolk syncytial layer, which is like an extra embryonic tissue that also expresses SOX32. But you can see that in the cells themselves, you see these hot cells, which go on to become the endoderm. So that's what specification looks like. And then at 9 hours, they're spread all over the animal. And if we take a sort of central slice through this confocal stack, you see that they are not at the surface, but they are underneath all of the other cells. So they're where they're supposed to be. If you take a look in an OEP embryo that has no nodal signaling, you still have SOX32 expressed in the YSL, but you don't have any cells that express it. And then if you look at later on in development, again, you don't see any, and so they can't be actually under any other cells because there aren't any. And then if we project light into our OEP embryos that have been injected with optoNodal, you see that we rescue endodermal specification. So we do get this scattering of endodermal cells. And then if you look later, they have spread over the embryo and they have gone to where they're supposed to be. So they are under the rest of the other cells. And so with this, really simple optogenetic stimulation, just kind of blasting the margin with light for about 2 hours, we can induce the right cells, and also they seem to do the right things after they're induced. And then, so this is the last figure in our paper, and the goal, we got to the experiment that was really the goal all along, which was to ask whether we can, to what extent can we replace nodal signaling with light at the phenotypic level, like at the, the phenotypic level of a full fish at 24 hours. So again, the experiment is we know what a wild type cell or embryo looks like at 24 hours. We know what an embryo that doesn't have any nodal signaling looks like at 24 hours. They're very unhappy and don't have any endoderm or mesoderm. And the question is, if we try to put that signaling back into the loss of function mutants, how close can we get to a wild type embryo with something really simple? And so this was really our first stab at this, and we're getting more sophisticated with the way that we're doing it now. But the idea was to leverage the ultra-widefield nature of the stimulation platform to explore not just 1 or 2 patterns, but spaces of patterns. And so from left to right, what you're going to see is we're illuminating from the top with either a thin donut or a thick donut. So we're not projecting a gradient, just like a band of signaling. And then along the vertical axis, we're trying dim patterns at the bottom, intermediate in the middle, and then strong on the top. And so we can really see kind of like how the phenotypes vary across this like phase space of different patterns. And so these are the embryos in the array, and you can see the actual pattern superimposed on top of them. And so the fun thing is that if you get an embryo that's kind of just right, so the ones that worked best tended to be bright donuts that were thin, you can rescue a lot of nodal dependent patterns. So this guy actually had a centrally located beating heart. It's got its trunk muscle back. You can see in the middle, you get the embryonic backbone, that's the notochord back. And a really cool one that we didn't expect was you actually get partial rescue of cyclopia. So if there are any connoisseurs in the audience, a characteristic feature of nodal mutants is that they only have one eye. And this guy has, the single eye has split into 2, although they are still touching a little bit. But so we're able to rescue a lot of the classic deficits that are associated with nodal loss of function mutants. If the signaling is a little bit too dim, we see something that looks like a nodal loss of function mutant. So it's kind of intermediate, but looks like somebody who needs more nodal signaling. And then if we try one of the really strong, thick patterns, we see that we can actually overdose them on nodal. And so this actually looks like a lefty 1-2 double mutant. So this is sort of characteristic of nodal hyperactive signaling. And so, if we, by tailoring the geometric characteristics of these patterns, we can really run the phenotypic spectrum from nodal loss of function to something approximating a rescue to nodal gain of function mutants. And just to close the loop, this isn't, just evident at the level of sort of gross transmitted light phenotypes, but we can also visualize markers of these nodal dependent tissues. So in a wild type embryo, you can see on the top in green is the notochord. And then the hatching gland, which is a sort of high intensity nodal target tissue. And then on the bottom, you can again see the hatching gland, but in green are the somites, which are the trunk muscle. MZOEP mutants have no nodal, so they don't have any of those things. But if we try to rescue our MZOEP mutants with targeted nodal stimulation, we can rescue at a molecular level the formation of the notochord, the somites, or the hatching gland. Okay, so that's basically everything I wanted to share with you guys. I think we're finally at a place where we can start to evaluate this original model where we have a gradient that gives rise to the endoderm, the ectoderm, and the mesoderm, and actually ask to what extent is that actually required for development, and to what extent does that maybe just reflect constraint? Like maybe it's totally possible that diffusion is a very convenient transport mechanism for molecules, and ultimately that's why you have a gradient. Because if you're using diffusion, it's kind of hard not to have one. You'd have to really go above and beyond to have something that isn't a gradient. And so maybe we're going to discover that actually you can rescue development with like a whole bunch of different flexible patterns, and we can finally start to ask, what is necessary and what is not in the context of these patterns. And I just want to highlight that this was recently published in our lab's first paper on bioRxiv. Please do check that out. Shout out to Vicente. He was super helpful in the development of this work, and also want to acknowledge that he was on the paper. And also thanks to everyone who's involved. So thanks to my postdoc mentor, Alex Shear, Adam Card and Catherine Rogers were both students in the Shear lab when I was there who helped with the embryo work. The Optogenetics is really the product of a long-term, super fun collaboration with Adam Cohen and his student, Harry McNamara, who's about to start his lab at Yale. And also, I forgot to put him on here, but yeah, thank you very much to Vicente for his contributions to the paper. Please do check us out. We're at the University of Pittsburgh. And also, just before I finish, I want to highlight that we are here at the University of Pittsburgh. If anyone is ever in town, come visit us. But also, I want to plug our PhD program. We have a joint PhD program with Carnegie Mellon University, which is also at Pitt. It's called the Computational Biology Program. So if that is your bag, please do reach out to us. It's a great program. But also I want to highlight that there are a lot of experimentalists here. I'm a part of this program, and I'm very much an experimentalist. And so if you are a quantitatively inclined biologist, please do check us out if you're looking for a PhD. We would love to have an application. So with that, I would be happy to take questions.
Excellent. Super interesting. Thanks so much, Nate, and thanks everybody for your questions. So I think we've been here for a while, and we'll have this seminar online. If anyone has more interest or more questions for Nate, please contact him or get in contact with us. I guess, I don't know. Thanks a lot. This was very, very nice.
Thanks everyone. Thanks for the invitation.